we've been looking at how the body senses itself, interoception. Now let's zoom in on hormones, specifically androgens, and their role in aggression. We often see stark differences between male and female brains in mammals, but not always. Take hyenas and naked mole rats. They're kind of sex-reversed, meaning females have higher levels of androgens, like testosterone. And guess what? These female hyenas are fiercely aggressive, challenging for dominance. This leads to a simple hypothesis. Maybe the reason typical females in most species aren't constantly brawling is simply because their androgen levels are lower. But hold on, life isn't that simple. Reality throws curveballs. You find species like the Brazilian guinea pig where the ladies have sky-high androgen levels, yet they're not exactly known for kicking male butt or taking names. Conversely, some sex-reversed birds don't even show elevated female androgen levels. It gets even more complicated when you look within a species. Just like with men, measuring a woman's androgen level doesn't reliably tell you how aggressive she'll be. And here's the kicker. Studies show that androgen levels don't typically spike right around the time a female is being aggressive. So that neat little cause and effect relationship starts to unravel. Why this complexity? Well, think about why females would ever need aggression. It's mostly tied to survival and reproduction, defending babies, competing for mates, fighting for resources needed during pregnancy or nursing. But here's the rub. Androgens, while potentially helpful for aggression, can mess with reproductive processes and maternal behaviors. Evolution, being the brilliant strategist it is, needed a way out. Females evolved a clever system, as Hurdy pointed out, to get the benefits of androgens for aggression without the downsides to having babies. It's like having a dimmer switch for androgens. They seem to selectively target the brain circuits involved in aggression, while leaving the reproductive and maternal circuits relatively untouched. Pretty sophisticated, huh? Okay, shifting gears slightly. Let's talk about something many women experience, PMS, or premenstrual syndrome. We all know the constellation of symptoms, mood swings, irritability, bloating, cramps, the works. For some, it's mild discomfort. For others, it's PMDD, premenstrual dysphoric disorder, where symptoms are severe enough to really mess with daily life, affecting about 2 to 5% of women. Now this topic is notoriously tricky, mired in two big controversies. First, what actually causes these symptoms? Is it purely biological, or is it something else? Second, and related to our interests, how much does PMS actually relate to aggression? Let's unpack these. The first controversy dives deep into the social construct camp. The extreme view, famously kicked off by Margaret Mead's observations in Samoa back in 1928, suggested that PMS is basically a Western invention. Her idea was that Samoan women didn't have these mood changes, implying it was due to cultural factors. This led to some rather dramatic interpretations blaming American capitalism, sexual repression, and general societal oppression for women's PMS. The argument went further, suggesting that women with bad PMS were inherently anxious, neurotic, repressed, compliant, withdrawn, and definitely not cool like the Samoans. Thankfully, this rather judgmental perspective has largely faded away. So, if it's not just culture, what is it? Well, science paints a different picture. Numerous studies have shown that there are indeed normal fluctuations in brain function and behavior throughout the menstrual cycle. Think about it. There are behavioral shifts linked to ovulation too. PMS then seems to be an amplified version of these natural rhythms. While the core experience is real, the specific symptoms can vary. Interestingly, women in China report less negative affect around their period compared to Western women, raising questions about whether they feel less, report less, or perhaps different symptoms altogether. And crucially, this isn't just human stuff. Both baboons and tamarins get more aggressive and socially withdrawn before their periods, suggesting a biological basis. What is socially constructed, perhaps, is labeling these normal variations as a disorder. Okay, so we know it's real, but what's the nuts and bolts biology? There are a few theories floating around. One popular one points to progesterone. As menstruation approaches, progesterone levels plummet, and this theory suggests we lose the hormone's calming effects. Sounds plausible, but honestly, the evidence isn't super strong. Another contender involves beta-endorphin, the same stuff that gives you that runner's high. This model proposes that PMS might be about abnormally low levels of this feel-good chemical. There's some support for this one, but let's be honest, there are plenty more theories out there, and frankly, we're still scratching the surface. The exact mechanism remains elusive. Now the million-dollar question, does PMS make women aggressive? Back in the 1960s, Katharina Dalton, who coined the term premenstrual syndrome, reported some studies suggesting female criminals committed more crimes during their perimenstrual phase. Another study of boarding school students found more behavioral infractions around that time. Sounds like a smoking gun, right? Well, not quite. The prison studies didn't differentiate between violent and non-violent offenses. And the school study lumped aggressive acts together with things like being late. When you dig deeper, the collective evidence doesn't really support the idea that women generally become more aggressive around their period, or that violent women are specifically primed to commit violent acts then. The link is tenuous at best. Despite the weak general link, there's a fascinating wrinkle involving the courtroom. Yes, defense attorneys have successfully used PMS as a plea for diminished responsibility. A really striking case from 1980 involved Sandy Craddock, a woman with a long criminal history including murder. She was a meticulous diarist, recording her periods and her criminal activities. Guess what? Her diary showed a remarkably close match between her menstrual cycle and her criminal sprees. Based on this, she received probation and treatment with progesterone. And here's the bizarre part. When her doctor reduced the progesterone dose, she was arrested shortly after her next period for attempted assault. Progesterone levels increased again, and presumably, so did her probation. What does this tell us? It suggests that for a tiny minority, PMS might trigger genuinely psychotic behavior, potentially offering a mitigating factor in court. But for the vast majority experiencing normal PMS fluctuations, increased aggression is not the norm. Alright, let's pivot to another major player in our internal landscape: stress. Often the moments leading up to significant actions, good or bad, are filled with it. And stress, unfortunately, tends to cloud our judgment, rarely steering us towards optimal decisions. Let's explore how stress works, particularly when it lingers, and how it messes with our brains. Remember ninth grade biology? Homeostasis, keeping your body in a stable state. A stressor is anything that throws that balance off, like a lion chasing a zebra. The stress response is the body's emergency plan. Two key brain regions kickstart this. First, the amygdala, the alarm bell. Seeing the lion activates the amygdala, which triggers the sympathetic nervous system, releasing adrenaline and noradrenaline for that immediate fight or flight surge. Simultaneously, the amygdala also signals the hypothalamus, specifically the paraventricular nucleus (PVN). The PVN then releases corticotropin-releasing hormone (CRH), which cascades through the pituitary gland and adrenal glands, ultimately releasing glucocorticoids (GCs) like cortisol. These two pathways work together. This acute stress response, the fight-or-flight system, is incredibly adaptive for surviving immediate physical threats. Think sprinting away from a predator. It rapidly dumps energy stored in your body into your bloodstream for your muscles. Heart rate and blood pressure shoot up to deliver that fuel faster. Crucially, it puts long-term projects like tissue repair and reproduction on hold, because right now, dodging that lion is priority number one. Plus, you get a natural painkiller boost from beta endorphins. Your immune system gets a temporary boost, clotting factors increase, and your brain's cognitive functions and senses get a sharpness upgrade thanks to glucocorticoids. This whole package is ancient, found across vertebrates. It's designed to save your bacon in seconds to minutes. But here's where modern life throws a wrench into the works. Primates, especially humans, have evolved this amazing capacity for anticipatory stress. We worry about future threats, social anxieties, financial pressures long before they materialize. If this stress response is constantly activated for psychological reasons, even when there's no actual lion chasing us, the system wasn't designed for this. The constant energy mobilization can lead to metabolic problems like diabetes. Constant high blood pressure leads to hypertension. Chronic suppression of growth and repair takes a toll. Reproductive hormones get disrupted, and while acute stress boosts immunity temporarily, chronic stress suppresses it. See the dichotomy? Acute stress saves you. Chronic stress, often driven by psychological load, actively damages your health. Before we dive deeper into the dark side of sustained stress, let's take a quick detour. Not all stress is bad, right? We actually seek out certain kinds of stress. Think about a roller coaster ride. It's stressful, your heart's pounding, you're scared you'll fall off. But it's fun. Why? Because it's mild, transient, and happens in a safe context. We willingly relinquish control and predictability for the thrill of the unexpected. This is the kind of stress we love: engaging, challenging, stimulating. It's the sweet spot of the inverted U curve. Too little stress is boring. Too much stress is overwhelming. There's an optimal range where stress enhances brain function, like boosting dopamine release via glucocorticoids. Rats will even work to self-administer the right amount of stress hormones. We crave that stimulation. Okay, back to the less pleasant side of sustained stress. So what happens when stress lingers? One major effect is on our fear circuitry, centered in the amygdala. Sustained stress makes us implicitly tune into angry faces more. That fast-track sensory shortcut from the thalamus straight to the amygdala becomes supercharged, prioritizing speed over accuracy. At the same time, the prefrontal cortex, our rational brain, gets quieter when processing emotions. The result? We become less accurate at quickly gauging someone's true emotion. Inside the amygdala itself, glucocorticoids play a big role. They flood the amygdala, making its neurons hyperexcitable, especially in the basolateral part BLA, which is key for learning fear. GCs don't create fear, they amplify existing signals. They also boost CRH and BDNF in the BLA, helping to solidify those fearful memories. Stress essentially makes it easier to learn and remember fears. This creates a nasty positive feedback loop. Stress ramps up the amygdala, which then ramps up the stress hormones, which then ramp up the amygdala even more. But here's another twist. Stress makes it harder to unlearn fear. Normally, the prefrontal cortex can inhibit the amygdala to help extinguish a fear response, like realizing a light no longer means shock. But stress weakens the PFC's grip on the amygdala. Fear extinction isn't just forgetting, it's actively learning that the threat is gone. So, sustained stress makes us better at learning new fears, but worse at letting go of old ones. Imagine learning to fear spiders easily, but struggling to ever feel safe again once you've learned that fear. Beyond fear, sustained stress wreaks havoc on our executive functions, the stuff handled by the frontal cortex. Working memory takes a hit, sometimes dropping to levels seen after frontal damage. High GC levels flood the PFC with norepinephrine, causing mental chaos instead of focus, and amplify disruptive signals from the overactive amygdala. Shifting attention between tasks becomes difficult. We become perseverative, stuck in ruts, doing the same thing over and over, even when it's not working. Stress weakens connections between the frontal cortex and the hippocampus, which is needed to learn from mistakes and change strategy, while strengthening connections to habitual circuits. This alters risk assessment too. Under stress, people might shift from being cautious to seeking big rewards, even irrationally. There's also a gender nuance. Major stressors push both sexes towards risk-taking, while moderate stressors tend to push men towards risk and women away from it, likely amplifying pre-existing tendencies. Bottom line, sustained stress messes up our ability to think clearly, make good decisions, and assess risks properly. Let's summarize the storm brewing in the stressed brain: an overactive, less accurate amygdala, dominating the hippocampus, and a muddled, impulsive frontal cortex. This sounds like a recipe for trouble, specifically reactive aggression. And indeed, studies show that stress and giving people acute doses of glucocorticoids increase aggression in both animals and humans. But there are important caveats. Stress doesn't create aggression out of thin air. It makes you more sensitive to triggers. And it hits hardest those already predisposed to aggression. There's also displacement aggression, lashing out at an innocent third party to relieve stress. This is surprisingly common, seen in baboons, and tragically mirrored in humans. Think increased domestic violence during economic hardship, or, chillingly, studies showing a spike in male partner violence after local football teams unexpectedly lose games. Perhaps the brain finds a grim reward in aggression, dampening the stress response itself. Stress doesn't just foster aggression. It also nudges us towards selfishness and away from empathy. One study showed that stressed participants were more likely to give egoistic answers in morally intense, personal scenarios, like the dilemma about leaving your family for love, especially if their glucocorticoid levels rose. It didn't affect milder dilemmas. This suggests stress makes us more self-centered, but only in the most emotionally charged personal situations, a pattern similar to frontal damage. And empathy? It seems stress can blunt that too. A fascinating mouse study showed mice lower their pain threshold when near a cadgemate in pain, but not a stranger. Blocking glucocorticoids allowed them to empathize with strangers too. It seems GCs narrow our circle of empathy. Similar results in humans suggest stress reduces our capacity to feel for others, especially those outside our immediate group. Pretty sobering stuff. Now, amidst all this talk of stress leading to negativity, Shelley Taylor proposed a counterpoint: the tend and befriend response, primarily observed in females. While males often default to fight or flight, females, according to this view, are more likely to care for offspring and seek social support when stressed. This fits with observed sex differences in coping styles. It likely reflects a stronger oxytocin release alongside the stress hormones in females. Of course, it's not a strict rule. Males can be prosocial under stress, and females can absolutely be aggressive. Think Gandhi or Sarah Palin. These exceptions highlight that individual differences and context matter immensely. Why these exceptions exist is a fascinating puzzle explored further in the field. It's crucial to remember that stress impairs judgment and decision-making, but its effects are value-neutral from a biological standpoint. When we say stress is adverse, we mean it disrupts optimal functioning. Consider an EMT becoming rigid and perseverative during a chaotic rescue. That's clearly bad for saving lives. But imagine a sociopathic warlord becoming equally perseverative during ethnic cleansing. From his warped perspective, it might be effective. Stress doesn't inherently make choices bad. It simply impairs our ability to make the best possible choice given the circumstances. It hijacks our cognitive machinery. Let's quickly tackle a common misconception: alcohol and aggression. The popular narrative is that alcohol lowers inhibitions, making people punchy. Wrong! Alcohol only reliably evokes aggression in two groups: those already prone to aggression. Think individuals with certain genetic predispositions like specific serotonin or oxytocin receptor variants, and those who believe alcohol makes them aggressive. Social learning powerfully shapes biology. For everyone else, alcohol usually leads to disinhibition, poor judgment, and maybe a drunken stupor that results in questionable decisions like impulsive Vegas weddings. So, I was drinking isn't a great excuse for aggression unless you fit into those specific categories. Hormones are powerful players, often having broader and longer-lasting effects than neurotransmitters. Take testosterone and aggression. Contrary to popular belief, it plays a much smaller role than assumed. Normal testosterone levels don't predict who'll be aggressive. Instead, it's facilitatory. Testosterone makes you more sensitive to triggers, especially if you're already inclined towards aggression. And during status challenges, rising testosterone might actually increase whatever helps maintain status, which could be prosocial behavior if status is earned that way. Oxytocin and vasopressin, they foster bonding, reduce anxiety, enhance trust, but with a huge caveat: they promote prosociality towards our in-group, us, making us more xenophobic towards outsiders, thems. They're parochial, not universal love hormones. And remember, female aggression, like defending offspring, is adaptive and facilitated by various hormones, including androgens acting selectively in the brain. So, let's quickly recap some key takeaways from this section. First, PMS is a real biological phenomenon, a variation of normal cyclical changes, though pathologizing it is a social construct. Its link to aggression is minimal except in rare extreme cases. Second, sustained stress is bad news for the brain. It ramps up the amygdala, shuts down the frontal cortex, reduces empathy, makes us more prone to aggression, and messes up our judgment. Third, hormones are complex facilitators, not simple determinants. They tweak our sensitivity to social cues and amplify existing tendencies, rather than creating behavior from scratch. Got it? Good. We've seen how hormones acting over hours can shape neural sensitivity right before an action. But what about longer timescales? What happens over days, weeks, or months? How do experiences stretching back this far influence our behavior? This brings us to the realm of neuroplasticity, the brain's remarkable ability to change its structure and function. We know short-term changes happen, like testosterone tweaking amygdala excitability. But over longer periods, the changes can be much more profound, reshaping the very architecture of the brain. Let's explore how events from months ago can still cast a long shadow on today's actions. How does the brain even remember anything? Early 20th century neuroscientists had some wild ideas. Maybe memory is stored in one neuron, so you need a new neuron for a new memory. Then maybe a neuron grows a new branch. Later maybe a new synapse forms, all discarded. The real breakthrough came in 1949 with Donald Hebb. His revolutionary idea, still fundamental today, was that memory formation doesn't require building new connections. It requires strengthening the ones we already have. His famous rule, neurons that fire together wire together. Basically, whenever two neurons fire together repeatedly, the connection between them gets stronger. Think of it like tightening a loose wire connection. The signal passes more easily. How does this strengthening actually happen at the molecular level? A key player is the neurotransmitter glutamate, the main excitatory messenger in the brain. Most glutamate receptors work simply. More glutamate binds, more sodium flows in, bigger excitatory blip. But some, called NMDA receptors, are trickier. They're usually blocked. They only open up and let a flood of sodium and calcium in when they receive a sustained barrage of glutamate, thanks to the non-NMDA receptors firing repeatedly first. It's like needing a warm-up before the main event. This threshold mechanism is crucial for learning. That lecturer droning on repeatedly, that's the repetitive glutamate release. The moment the dendritic spine finally gets it and the NMDA receptors activate, that's the light bulb moment, the initial strengthening of the synapse. Okay, so the NMDA receptors fire, the calcium rushes in. But how do we make this potentiated state last? That's where long-term potentiation, or LTP, comes in. Discovered in 1966, LTP is the process that turns that initial burst of activity into a lasting long-term increase in synaptic strength. The magic ingredient is calcium. When calcium floods in through the NMDA channels, it triggers a cascade of changes. It causes more glutamate receptors to be inserted into the postsynaptic membrane, making the neuron more responsive. It modifies existing receptors, making them more sensitive. And it even sends signals backward to the presynaptic neuron, telling it to release more glutamate in the future. It's a combination of the sender shouting louder and the receiver listening better, creating a stable, long-lasting connection. This is the cellular basis for memory consolidation. If there's LTP, there must be LTD, long-term depression. Just as LTP strengthens synapses, LT weakens them. It's experience-dependent, making the synapse less excitable over the long haul. Now, don't think of LTD as just generic forgetting. It's thought to play a crucial role in sharpening neural signals by selectively erasing irrelevant information. It's like decluttering your mental workspace. Importantly, LTD doesn't just work by reversing LTP. It uses different molecular pathways. Together, LTP and LTD provide a dynamic balance, constantly sculpting our neural circuits based on what we experience, strengthening useful connections and weakening noisy ones. For a long time, LTP was heavily associated with the hippocampus, the seat of explicit memory, like remembering phone numbers. But guess what? LTP isn't exclusive to the hippocampus. It happens throughout the nervous system. In the amygdala, LTP underlies fear conditioning, learning to associate a bell with a shock. In the frontal cortex, it's involved in learning impulse control. Even dopamine systems use LTP to associate cues with rewards, like how addicts link places to cravings. Now, add stress hormones back into the picture. Moderate stress can boost LTP in the hippocampus, while prolonged stress messes it up, promoting LTD, explaining why cognition tanks under chronic stress. In the amygdala, sustained stress cranks up LTP and dials down LTD, making it easier to learn fear and harder to unlearn it. In the frontal cortex, sustained stress suppresses LTP, impairing learning new control strategies. Put it all together, stronger fear circuits, weaker control circuits, and you get a recipe for stress-induced impulsivity and poor emotional regulation. Remember how Hebb's idea of strengthening existing synapses dominated the field? Well, ironically, the discarded idea of forming new synapses has made a comeback. Techniques for counting synapses show that enriching environments actually increase the number of synapses in the rat hippocampus. Using fancy imaging, scientists can literally watch new dendritic spines sprout on a neuron's branch, followed by an axon terminal hovering nearby, eventually forming a new functional synapse. This activity-dependent synaptogenesis can even be triggered by the calcium influx during LTP. New synapses form when you learn new motor skills, after lots of visual input, or when whiskers are stimulated. Enough new synapses can even cause the entire dendritic tree to expand. So, it's not just about strengthening old wires, but sometimes laying down entirely new ones. And guess what? Stress hormones have that familiar inverted U-shape effect on synaptogenesis too. Moderate transient stress, or equivalent glucocorticoid levels, can actually increase the number of new spines in the hippocampus. But sustained stress or high GC levels do the opposite, reducing spine number and dendritic complexity, particularly in the hippocampus and frontal cortex. This ties into depression and anxiety, which are linked to high GCs and reduced hippocampal structure, possibly due to lower levels of the growth factor BDNF. In the frontal cortex, sustained stress leads to synapse loss and impaired function. Interestingly, the amygdala behaves differently under sustained stress, increasing BDNF and expanding dendrites, which fuels anxiety and fear. There's also a fascinating context dependency. The same GC levels can cause dendritic growth if the amygdala is also active. Good stress, like voluntary running, but atrophy if it's just the stress hormone surcharge, bad stress like fear. And hormones like estrogen also play a role, increasing spine number and cognitive performance during high estrogen phases. Plasticity isn't just about synapses and dendrites. Axons, the output wires of neurons, can also sprout new branches and forge new connections. This is called axonal sprouting. The most spectacular examples come from sensory substitution. Blind people reading Braille activate both their tactile cortex and, remarkably, their visual cortex. How? The neurons normally sending touch signals to the fingertip area of the somatosensory cortex have grown new projections miles away to the visual cortex. A blind woman who had a stroke in her visual cortex lost her ability to read Braille. Blind subjects trained to associate letters with sounds activate their visual cortex when processing those sounds. Similarly, deaf people fluent in sign language activate their auditory cortex when watching signing. This remapping isn't just for injury recovery. After a stroke-damaging hand sensation, axons from nearby wrist receptors can sprout into the vacant cortical territory, slowly restoring some touch sensation. How do these axons find empty space, figure out what to connect to, and navigate such long distances? That's still a hot research area. This kind of remapping isn't just for injury or blindness. It happens constantly in healthy brains too. Musicians, for example, have larger auditory cortical areas dedicated to musical sounds, especially their own instrument. The earlier you start playing, the stronger this remapping. Short-term training can induce rapid, though temporary, remapping. Practice a piano exercise for 2 hours a day for a few days, and the motor cortex controlling that hand expands, likely due to transient Hebbian strengthening. Do it for 4 weeks, and the expansion becomes lasting, probably involving new axonal sprouts and connections. Even imagining playing the exercise causes remapping. Other examples abound. Postpartum rats show expanded cortical maps for nipple touch, and jugglers develop larger cortical areas for processing visual motion. The takeaway? Experience constantly reshapes the brain's wiring, changing synapsina numbers, dendritic trees, and even where axons project. Now let's talk about perhaps the biggest revolution in neuroscience in decades, adult neurogenesis. For ages, the dogma was clear. You're born with all the neurons you'll ever have, and it's downhill from there. New neurons? Forget about it. But then, in 1965, Joseph Altman, using a novel technique to label newly formed DNA, found evidence of new neurons being born in the adult rat brain. Initially, it was exciting, but soon the field turned hostile. Altman faced rejection, couldn't get tenure, and lost funding for this work. A decade later, Michael Kaplan tried to replicate and extend Altman's findings with better techniques. Again, mostly crushing rejection. Even prominent figures like Pasco Rakic publicly dismissed Kaplan's work, famously telling him, "Those may look like neurons in New Mexico, but they don't in New Haven." Kaplan eventually left research. The field was stuck. Silence reigned until Fernando Nottebohm, studying birdsong, provided compelling evidence in the 1980s. Birds that learned new songs each year were making new neurons in their brains. Nottebohm's prestige lent credibility, shifting the debate from, "Does it happen?" to, "Is it relevant in mammals?" Then, in the 90s, Elizabeth Gould and Fred Gage used even more sophisticated techniques to convincingly demonstrate robust neurogenesis in rodents, including in the cortex. Rakic eventually came around, but with skepticism. Okay, it happens, but only a few neurons, they don't live long, it's not in the important areas, and it's only in mice. Soon after, it was shown in monkeys, then humans. Crucially, it was proven that these new neurons integrate into existing circuits, become electrically active like young neurons, and are essential for pattern separation, the ability to distinguish between similar things, like telling dolphins and porpoises apart. This is huge. So why did it take so long for this revolutionary idea to be accepted? There are different takes. Some argue the skeptics provided necessary quality control, pointing out flaws in early work. Others paint a picture of the old guard clinging stubbornly to dogma, despite the evidence. Rakic's famous quote certainly doesn't help that narrative, though Altman wasn't exactly a young rebel. Joseph Altman himself reflected on this in his memoir, expressing confusion about the initial excitement followed by rejection, perhaps wondering if he focused too much on lab work and not enough on marketing his discovery. He maintained a philosophical detachment, having escaped a Nazi camp. Whatever the reasons, the field exploded once acceptance dawned. Adult neurogenesis is now the hottest topic in neuroscience, with thousands of papers citing it annually, exploring everything from how exercise stimulates it to its potential role in depression and antidepressant action. So, wrapping up this section, we've journeyed from the immediate hormonal whispers before an action to the profound ways experiences over days and months reshape our brains. We've seen how the brain isn't fixed, but constantly molded by hormones, by strengthening and weakening connections, by forming new synapses and remapping axons, and even by birthing entirely new neurons. It's a dynamic, ever-changing organ, reflecting the complex interplay of our past shaping our present actions.